So, I want you to imagine something for a second. Picture yourself sitting in a doctor's office. You've been dealing with this mysterious, really debilitating illness for years.
Right.
And you finally decided to just go ahead and get your entire DNA sequenced to find the root cause. So you're sitting there on the exam table, holding your breath, waiting for an answer, and the doctor walks in, looks at your chart, and says, well, we found a mutation in your genetic code, but we have absolutely no idea what it does.
Yeah, that is an incredibly frustrating scenario. And unfortunately, it plays out all the time in modern medicine. Geneticists refer to this as the variant of uncertain significance, or VUS problem.
The VUS problem.
Exactly. It remains arguably the single most massive bottleneck in clinical genetics today.
Which perfectly brings us to the mission of today's deep dive. We are going to explore how scientists are, finally cracking this dark matter of our genetic code.
It's about time, honestly.
It really is. We're looking at two groundbreaking sets of research today that approach this massive problem from wildly different angles. One strategy uses just massive biological brute force, and the other takes a completely different path, looking deep into the evolutionary history of primates.
Which is fascinating.
And combining that with cutting-edge 3D artificial intelligence.
To give you some context on the sheer scale of the problem we're dealing with here, there are over 70 million possible protein-altering variants in the human genome.
Wow. 70 million.
70 million. And our current clinical databases only actually understand the effects of about 0.1% of them.
So the other 99.9% are just a complete mystery.
A total black box.
Yeah.
And the research we're dissecting today really matters because it provides a shortcut to actually reading the grammar of our DNA. We're finally getting the tools to filter out all that endless background noise and pinpoint the exact specific mutations that cause disease.
Okay, let's unpack this. Starting with the first approach, which was spearheaded by a researcher named Siraj and their team. To really understand what they did, we have to look at how we've been hunting for disease genes up until now.
Right, the old methods.
Yeah. For a long time, the gold standard has been something called genome-wide association studies, or GWAS. And GWAS is fantastic at finding the general neighborhood of a disease-causing gene.
It gets you to the right zip code.
Exactly. It can tell you the mutation causing a disease is somewhere on a specific block. But the problem is, it's terrible at finding the exact house on that block. And that is mostly because these variants are hiding out in the non-coding regions of our DNA.
Yeah, and the non-coding regions are the parts of our DNA that don't directly build proteins. Instead, they act as the control switches, turning genes on and off. Finding a broken switch in, millions of base pairs is incredibly difficult.
Like finding a faulty wire in a skyscraper.
That's a great way to put it. So to solve this, the researchers used a technique called MPRA, which stands for Massively Parallel Reporter Assay. You can think of it like a massive biological audition.
An audition for hundreds of thousands of actors all at once. I mean, how do you even organize a test like that?
It's wild. The scientists took 221,412 of these fine-mapped variants. These are the suspects that GWs had narrowed down to the right neighborhood. They synthesized them into over 300,000 unique pieces of DNA.
That is an insane amount of synthesis.
It is. And they attached a tiny, unique molecular barcode to each one. Then, they basically dropped these DNA elements into 5 completely different types of human cells growing in a dish.
Right.
We're talking about liver cells, brain cells, blood cells, lung, and colon cells.
So they essentially tossed all these genetic suspects into different cellular environments to see what they would do. But how do those barcodes actually help them measure the results?
By reading those tiny barcodes, the researchers could measure exactly how much RNA output each specific piece of DNA was generating. It allowed them to see which mutations were physically altering the behavior of the cell.
And which ones were doing nothing.
Exactly. Which ones were just harmless background noise. Out of all those hundreds of thousands of suspects, they identified 13,121 high-confidence variants. They call these TARVs, or trait-associated regulatory variants. They successfully separated the actual functional signals from the noise of the genome.
Okay, here's where it gets really interesting. They didn't stop at looking at single variants in isolation, because genetics isn't always a solo act. Sometimes variants interact with each other in a phenomenon called epistasis.
Yes, epistasis is huge.
They started testing pairs of variants together to see if the combined effect was different from just adding their individual effects. And they found these non-additive epistatic effects in 11% of the pairs they tested.
What's really fascinating here is how varied those interactions can be. Take the ESS2 locus, for example. It's a region linked to developmental disorders like DiGeorge syndrome. They found 2 variants there that both independently activate transcription.
Which is just the process where the cell reads the DNA recipe to make an RNA copy, right?
Exactly. But when you put those 2 variants together, they amplify each other to create a massive effect, way beyond what you'd expect from just adding them up.
It sounds like pouring gasoline on a fire. Did they find instances where the variants behave differently, maybe shutting each other down instead of amplifying?
They did. At the THBS2 locus, which is linked to systolic blood pressure, they found 2 variants that had a totally different relationship. For the gene switch to turn on, both variants must be present simultaneously.
Like turning 2 keys to launch a missile.
Precisely. Specifically, both need to be the C allele. And an allele is simply a specific version or flavor of a genetic letter at a given spot. If you only have one C allele, nothing happens. You need both to physically create a functional gene motif.
Now, a motif, what does that mean in the context of DNA?
Think of a motif as a specific sequence pattern that acts like a landing pad. Cellular machinery needs to recognize that exact pattern to land on the DNA and activate the gene. If even one of those C alleles is missing, the landing pad is broken.
The machinery can't dock?
Right? The machinery can't dock and the gene stays off. It really shows how incredibly complex and nonlinear our genetic grammar actually is. It is not just about having a mutation. It is about the combination of mutations.
And to dig even deeper into that grammar, the researchers used a brute force technique called saturation mutagenesis. They took 128 of these genetic elements and just methodically mutated every single letter one by one to map out exactly what happens to the cell's output.
It is a phenomenal amount of work, but the payoff is huge. It leads to real breakthroughs in understanding context. One compelling example from the paper involves a gene called CDHR3.
Oh, this is the liver cell one.
Yes. They found a specific variant that creates a binding motif called SRYSOX9. But the catch is that it only created this effect in HepG2 liver cells. And the SRY protein is Y chromosome linked.
Meaning it's tied to biological sex?
Exactly. That means this specific genetic variant has sex-dependent effects. It actively drives higher expression of the gene in XY individuals compared to XX individuals.
That is wild. A mutation might be completely silent in half the population and actively driving gene expression in the other half, all because of the chromosomal environment it finds itself in. We really can't look at DNA as just the static string of text that behaves the same way in everybody, or even in every cell within the same body.
We can't. That limitation, the fact that context is absolutely everything, forces us to look for broader patterns. Because while the first set of researchers did a masterful job illuminating the non-coding dark matter, the control switches, we still have a massive challenge understanding the actual protein-coding genes.
The recipes themselves.
The recipes, yeah.
Because if you have a mutation right in the middle of a recipe for a crucial protein, how do you know if that mutation is going to ruin the recipe, or if it's just an alternative spelling that doesn't change the flavor at all?
It's the ultimate question.
Right. And to answer that, another group of researchers, Gao and colleagues, looked outside of humanity entirely. They turned to our closest living relatives, the primates.
The scale of this primate database is hard to overstate. They sequenced 809 individuals across 233 different non-human primate species.
233 species.
From lemurs to chimpanzees. And they cataloged 4.3 million common variants.
But why primates, specifically? I mean, why not use data from lab mice, which we already study so heavily in genetics?
It really comes down to our shared evolutionary history. Humans and chimpanzees share 99.4% of our protein sequence. It's nearly identical. So the researchers hypothesized an evolutionary shortcut. If a specific mutation is common in a wild primate population, and those primates are swinging through trees and surviving perfectly well in nature...
And it's probably fine for us.
Exactly. That exact same mutation is almost certainly harmless if it shows up in a human.
That makes intuitive sense. If it doesn't break a macaque's heart, it probably won't break a human's heart. Did the data actually back that up?
Emphatically. When they cross-referenced their primate database with human clinical databases, they found that 99% of these common primate variants perfectly match the benign label in humans.
99%? That's incredible.
It is an incredibly reliable mirror, and they tested this against other animals too, just to be sure. If you try this same trick with more distant mammals like mice or dogs, the accuracy drops to 87%.
Still okay, but not 99.
Right, and if you go all the way to chickens, it drops to 71%. Non-human primates really are the evolutionary sweet spot for understanding our own bodies.
That's a great transition, actually.
If we connect this to the bigger picture, this primate mirror doesn't just tell us what is harmless. It also shines a bright light on what makes us uniquely human. By comparing the genetic constraints of primates to humans, we can see exactly where our evolutionary paths diverged.
And the paper actually highlights genes that are under less evolutionary constraint in humans compared to primates, meaning mutations that might be weeded out by evolution in monkeys are somehow tolerated in us. They pointed to genes like CFTR, which is linked to cystic fibrosis, GJB2, linked to hereditary deafness, and CD36. Why would humans tolerate potentially deadly mutations more than a monkey would?
It all comes down to a concept called heterozygote advantage.
Okay, what is that?
Well, having two copies of these mutations causes the disease. But carrying just one copy of the mutation might actually provide a protective advantage against local environmental pathogens. Carrying one copy of certain mutations can protect local populations from devastating diseases like malaria or cholera.
Ah, so it's an evolutionary trade-off.
A huge one. The mutation stays in the human gene pool because historically it offered a survival benefit against those specific human-centric threats.
That explains why we might hold on to certain risky mutations. What about the flip side? Are there genes where human evolution is actually stricter than primate evolution?
Absolutely. The TERT gene is a prime example of this. It's responsible for maintaining telomeres, which are the protective caps on the ends of our chromosomes. TERT is under much tighter quality control in humans than in other primates.
Telomeres are usually associated with aging, right? Does this have to do with how long we live compared to other primates?
That is the most likely reason. The median lifespan of a non-human primate in the wild is roughly 20 to 30 years. Humans, on the other hand, have evolved to live much, much longer. Because our cells have to divide and replicate for 7 or 8 decades, we need significantly stricter maintenance of our telomeres. We simply can't afford the sloppy mutations there that a primate might easily survive with during its much shorter life.
So it's evolution tightening the screws on our DNA so we can live to see our grandchildren.
Beautifully put.
But having this massive database of 4.3 million benign primate variants was really only phase 1 for these researchers. Phase 2 is where they took all this primate data and used it to train a deep learning model called Primate AI 3D.
This is where the methodology truly leaps forward. Previous AI models tried to predict disease by looking at DNA as a linear string of text. But proteins aren't flat text. They fold into incredibly complex three-dimensional machines. The researchers took advantage of AlphaFold data to voxelize the 3D structures of human proteins.
Voxelize, I love that word. If you've ever played Minecraft, you know what a voxel is. It's a 3D pixel, a little block, and a 3D grid. They essentially broke these microscopic proteins down into 3D grids so the AI could analyze their physical shape instead of just reading their chemical spelling.
Then they used a technique called semi-supervised learning. Supervised learning is when you hand an AI a data set where every single item is labeled with the answer. Unsupervised learning is just dumping raw, unlabeled data and hoping the AI finds a pattern. Semi-supervised learning falls in the middle. They fed the AI the 3D protein structures along with those 4.5 million benign primate variants. They only taught the AI the shape of normal.
Wait, so they didn't explicitly teach the AI what a disease-causing mutation looks like at all?
They didn't have to. The AI learns where harmless variants tend to cluster on the protein surface. Crucially, it infers the rest by looking at the empty spaces. It figures out that the structural core of the protein where primate variants never appear must be critically important.
Because evolution protected it.
Exactly. If it sees a theoretical mutation happen in those empty spaces, the AI flags it as highly pathogenic, because evolution clearly never allowed a change there to survive.
That is a brilliant way to map the danger zones without needing to see the danger first. How did this 3D semi-supervised model actually perform against the tools doctors are using today?
They tested Primate AI 3D against 15 other major machine learning models. They ran it through 6 massive real-world clinical benchmarks, including the UK Biobank and databases for congenital heart disease and autism spectrum disorders. Primate AI 3D outperformed every single one of them.
The technical achievement is impressive, but does this actually translate to helping that patient sitting in the doctor's office waiting for answers?
The clinical payoff of this accuracy is the most important part of the research. By applying this AI to a cohort of patients with neurodevelopmental disorders, they were able to discover 290 candidate genes linked to intellectual disability.
Wow.
That is real, tangible progress. It means providing answers and diagnoses to families who have spent years caught in the agonizing limbo of that variant of uncertain significance label.
So what does this all mean for you? If you are that patient, why should you care about barcodes in a Petri dish, or the DNA of a lemur, or an AI that looks at proteins like a Minecraft grid?
It means we are finally moving away from educated guessing. Whether we are using massive biological assays to test hundreds of thousands of cell interactions in a dish, or looking at the DNA of 233 different primate species to train a 3D neural network, we are building empirical tools to decode our own biology. We finally have the biological and evolutionary compasses required to navigate the human genome. It means that in the very near future, personalized medicine will actually be precise and definitively mapped to your unique genetic makeup.
I want to leave you with one final, slightly provocative thought. Something hidden right at the very end of that second paper. The researchers note that this incredible AI, this tool that is currently diagnosing human diseases and finding candidate genes for intellectual disabilities, was built entirely on the genetic diversity of wild primates. But right now, more than 60% of the primate species on Earth are threatened with extinction in the next decade due to human activity. Their DNA just unlocked our ability to read our own health. You have to wonder, what other incredible medical blueprints, what other cures and insights are hiding in the genomes of species we might lose before we ever even get the chance to sequence them?
It is a sobering reminder that conservation isn't just about saving nature. It is very literally about preserving the library of our own biology.
Thank you for joining us on this journey through the dark matter of our DNA. We appreciate you coming on the deep dive with us. Catch you next time.